an ARIS group at the Australian National University, and today I'll be talking to you about RISER, a technology for the biochemical-free enrichment or depletion of RNA classes in real time during direct RNA sequencing. The transcriptome contains a diverse range of RNA classes which vary in their structure, function, and role in disease. These classes are not equally represented in cells, and it remains a major challenge to characterise RNA with low abundance, particularly in the presence of highly abundant RNA. For instance, long non-coding RNAs usually have a low but highly tissue-specific expression pattern, which can be obscured in RNA measurements as a result of the highly abundant mRNA. Similarly, in cardiac and other muscle samples, mitochondrial RNAs dominate, and this takes sequencing resources away from other transcripts. To enrich or deplete specific RNA molecules for sequencing, biochemical approaches can be used to alter the distribution of transcripts in the sample prior to sequencing. But these are time-consuming, expensive, risk degrading the RNA, and in turn this can limit the length of reads and bias their content. When these biochemical approaches are coupled with cDNA sequencing, there are further limitations introduced by reverse transcription, mainly the inability to study the epitranscriptome, as well as the introduction of nucleotide biases as a result of amplification. Nanopore sequencing allows the direct measurement of RNA without reverse transcription to cDNA. It measures individual RNA strands by recording a current signal while the strand moves through the pore, and this current signal can then be decoded to understand features of the RNA molecule, such as its nucleotide sequence or the presence of biochemical modifications. However, the throughput of nanopore technology is limited, and the pores degrade over time. So it's really important to focus the limited sequencing capacity on the RNAs of interest for the duration of the run. A unique advantage of nanopore technology is the adaptive sampling feature. So this is where in real time signals get base called and then mapped to a list of targets, and if the molecule being sequenced doesn't match the target, it can be selectively rejected from the pore by reversing the pore voltage. There are a couple of limitations with this. In particular, the targets need to be specific sequences that are known beforehand, and this is problematic if you're dealing with organisms with incompletely defined references. In addition, the base calling and mapping operations are computationally expensive, and this is problematic because you need to be making decisions as quickly as possible for adaptive sampling to be beneficial. And this is even more important for RNA, which typically has much shorter read lengths than for DNA. To overcome the limitations of these various targeted RNA sequencing approaches, we've developed RISER. RISER uses a neural network to classify RNA directly from the start of the raw nanopore signal, then sends an accept or reject command to the sequencing hardware depending on the user-defined target RNA class. This focuses the limited operating time of the nanopores only on the RNA class of interest. Since RNA is always sequenced in the 3' to 5' direction of the molecule, the start of the nanopore signals always correspond to the 3' end of RNA. The RISER decision therefore relies on being able to distinguish between the 3' ends of different RNA classes. As I mentioned earlier, mRNA is the most abundant class in standard direct RNA runs and occupies most of the sequencing capacity. This is a problem if you're interested in sequencing non-coding RNAs. And importantly, mRNA has distinct features in the 3' UTR, which are not present at the 3' end of non-coding RNAs. So we decided to use mRNA as our first target class to implement in RISER. Before selecting which model to use, we first needed to decide what signal length to use as input to RISER. This required a bit of a trade-off because if the input was too long, then we risk reads escaping through the pore before a decision can be made. But if the input is too short, then we might not have enough information to make a good prediction. So from this perspective, the best trade-off we found was 4 seconds. We then tested 3 different types of convolutional architectures to determine whether a signal is mRNA or not. Convolutional architectures are very useful for analysing nanopore signals because they can learn local patterns anywhere in the signal and with varying timescales, which suits the signal variance in these signals. We systematically optimised the hyperparameters of each of these architectures and then tested the best configuration for each one on a held-out testing dataset. For all 3 architectures, the accuracy was best at signal lengths of 4 and 5 seconds, and given the plot on the left, we thought the best trade-off was then to select an input of 4 seconds. This corresponds to around 280 nucleotides at the 3' end. This was also supported by the ratio of true positive rate to false positive rate, and so the deciding factor for which architecture to select became the time to make a prediction. Since this is a real-time application, we want to be making predictions as quickly as possible, and you can see that the CNN in blue is around twice as fast as both the ResNet and TCN, but also still accurate. Having selected the CNN model, we then evaluated its ability to generalise to new samples by testing on an independent dataset from HeLa cells, which hadn't been used for any training or hyperparameter tuning. For classifying between mRNA or not, RISER model got an accuracy of 88% and an area under the ROC curve of 0.96. We next ran RISER in real time during live MinION sequencing of poly-A-selected RNA from HeLa cells. 4 libraries were prepared to use in 4 runs. The first used RISER to enrich mRNA, the second used RISER to deplete mRNA, the third was a control without RISER, and then the fourth was a split flow cell where we used a single flow cell, split the channels into 3 groups to simultaneously test all 3 conditions in a single flow cell to reduce the effect of variability between flow cells. We also repeated the same set of experiments on a second cell line. We found that among the reads that RISER accepted through the pore, the target RNA class was enriched while the off-target class was depleted. When the goal was to enrich mRNA, the mRNA reads in pink increased and the non-coding or the non-mRNA reads in blue decreased, and vice versa when the goal was to deplete mRNA. The same enrichment trends were observed in all experiments. As I showed in the first slide, long non-coding RNAs make up a very small portion of reads in nanopore direct RNA runs, but they're biologically interesting, so we wanted to see the effect of RISER on these RNAs. So when depleting mRNA, which is the right-hand columns, we found that the long non-coding RNA proportion in light green increased by a factor of 2.2. And when enriching mRNA, the proportion of mitochondrial RNAs decreased by a factor of 33, indicating RISER might also be useful for that application. Again, these results were consistent across all experiments. Importantly, we observed that RISER did not impact the percentage of available pores after 24 hours of sequencing. This is important because voltage reversals for DNA adaptive sampling are thought to damage pores and reduce their working time, but we didn't find this to be the case in our direct RNA runs. We've developed, so the examples I've shown today are for the application of RISER to enrich or deplete mRNA, but the RISER software is designed to be modular and extensible and allow easy adaptation to different targets. We've also developed a simple command line tool to run RISER. You only need to specify your target class, whether you want to enrich or deplete the target class, and the duration to run RISER for. You don't need to change anything when setting up your run. RISER will just interact with the sequencer in the background. So to summarise, RISER is the first biochemical-free technology for real-time enrichment or depletion of classes of RNA during direct RNA sequencing. It alleviates the limitations of biochemical enrichment and also does not require any form of target reference to provide a level of sequencing control that's currently not possible with any biochemical or computational method. It's available now for RNA researchers to use to enrich or deplete mRNA, and more models are on their way. I'd really like to thank the rest of the RISER team, in particular Eduardo Ayres, Agan, Nikolai and Nadine, the rest of the Ayres group, and the support of Carolina at the Biomolecular Resource Facility at ANU. Thank you very much.